Hello everyone, thank you for stopping by at my poster presentation recording. My name is Rucheng Diao. I'm a PhD candidate at the University of Michigan. Today I'm going to talk about the project we've been working on integration of large-scale expression profile into a multi-method protein function prediction pipeline. So let's get started. So the overall goal of the project is that with all the information of protein out there, yet with unknown function, whether or not we can computationally predict what the function of proteins are. So there are a lot of computational methods developed. In our case, at University of Michigan, our collaborators lab and us has been working on the medical method, which is actually a pipeline that makes use of both structure and sequence similarities, plus protein-protein interaction data for function prediction. And part of medical has been submitted for Kappa-3. And speaking of Kappa-3, after the result of Kappa-3 and Kappa-Pi came out, we noticed that in the Pseudomonas example that I gave, the expression data is actually helping the performance a whole lot, even if a simple baseline implementation making use of expression data actually outperformed the top submitted methods in the baseline. So at this point, we started to wonder, we have sequence structure and protein-protein interaction information available in medical. If we supplement it with expression data, whether or not we can help the prediction based on the co-expression patterns. So the first thing we look up is whether or not there's actually enough data out there. Expression profile is actually available for a variety of species, either in data repositories that may have multiple organisms in it. And you can kind of visualize the expression profile like a heat map with samples on one axis and genes on the other axis. And besides that, there are also curated expression profile compendia that's out there. In our project, one of the extensively used one is the Columbus, which is one that focuses on microarray data for prokaryotic model. And again, the expression profile can kind of be visualized as a heat map. So here is our general method of how to incorporate expression profile information into predicting the function. So let's say we have a gene G on the left-hand side, and we are wondering what its function is. And because most of the expression profile is done on the transcriptional level, hence we are annotated as gene in this case. And what we can do is that consider another gene that we might have better idea of what its function is across different experiments out in the expression profile we can find out there, whether or not they co-express across different experiments. And it can be measured as some kind of correlation or mutual information in our case between the gene of interest and another gene in the organism. So we can repeat this process for a lot of other genes/protein in the organism, and each one of them, if annotated with function, will know what it is annotated with, and with the correlation of correlation score that measures co-expression between this gene of known function and the gene of interest. So across all the genes that's out there, we compile them into a final confidence score that is of the gene G, which is our gene of interest, against possible functions that has been annotated out there. So basically, we condense the information of gene G co-expressing with any possible other genes in this organism, and combine with their own annotation of the function that we already know to come up with a measure of how confident we are that gene of interest is related to a certain function. So that's the general thought of how the pipelines is going to work. And the implementation is going to be divided into 5 major parts. First of all, we need to get the expression data. And with that, we can calculate the co-expression scores between genes, just like we just described. And we calculate the confidence score for gene versus function after compiling different genes together. And we do the backpropagation of the hierarchy of the functions at the end of it. And we would like to benchmark and see how our method is doing. So that's the idea and the implementation of our method. And we actually did some benchmarking already with the data that's available. So we have a 1200 also entry of curated E. coli dataset that is pretty well annotated. And in our case, we focus on the FX measure for this specific project, and I'm going to show an E. coli case example in the following slide. So as you can see, there are a lot of cells on it. Basically, that's coming from a lot of different ways we can set up the parameters. So for example, the top half of the plot is the quote-unquote include mode, which has the information also put into calculation if we already know the gene of interest is annotated with a function of interest. But realistically, most of the time, we are probably going to encounter an exclude kind of version that we don't actually know a whole lot about this gene and this function. So as you can see, we have a method out here that is having a pretty high Fmax score. And it is actually a way of normalizing confidence score by calculating the sum of co-expression and then normalize it by only the top 20 of the co-expression score out there. And hopefully we can minimize the effect of noise in this case. And it looks like it's helping. And after wrapping up for Kappa 4 submission, we plan to continue the work to build a generalized method to implement expression profile into protein function prediction. So that's all for me for today, and here's our information again. Feel free to let us know if you have any questions.